Hello, welcome to another episode of Microbiology. So today we're going to be covering gram-positive, but it's a lot. So we're going to divide this video into two parts, part 1 and part 2. The goal is to tackle the higher points of when it comes to gram-positive bacteria. So in the next video, we will talk about gram-negatives, but this is exclusively on gram-positive bacteria. All right. So the first thing you want to think about, okay, is the peptidoglycan layer of the gram-positive. Compared to the gram-negative, it is very thick. The peptidoglycan layer of the gram-positive is very thick compared to the gram-negative. And there is one common feature among gram-positives that you never find in gram-negatives. And this is a very, very common exam question. And it's called teichoic acid. So teichoic acid is only found in gram-positives. You do not find it in gram-negatives. For example, so there will be a question on the exam, and then they will give you like unique features of, let's say, Staph aureus, which is a gram-positive, right? And then they ask you a question like, what are some of the things, or what is the common feature that you find in Staph aureus that you don't find in something like E. coli? And you know that E. coli is gram-negative, Staph aureus is gram-positive. So you find teichoic acid in gram-positive. So that should be your answer, right? All right. So now let's start. So the first thing we want to do is when we look at the shapes, okay, so we have rod shapes, we have the rod shape, and then we have cocci. So the first we want to talk about the cocci, right? So today is on gram-positive. Gram-positive. And remember, guys, when you stain gram-positive, they usually look blue or purple. They usually look blue or purple. And gram-negative is gonna be more like red or orange or something like that. All right, so we can divide this into like rod, and so we can have like rod shape, and then we have the cocci, which is COCCI. Okay. Our video today is mostly gonna be on this. So for the rods, we're gonna tackle them tomorrow. So for the rods, we know we have B. I have this mnemonic BLCC, right? Which is the B is for bacillus. Bacillus. So for bacillus, remember we have two types. We have bacillus anthracis. And then we have the bacillus cereus. All right, and then the L is for listeria. So listeria monocytogenes. So the first C is, we have Clostridium. And under Clostridium, we have so many. So under Clostridium, we have Clostridium or C. tetani, which causes tetanus, right? So we have C. botulinism, which releases the botulinum toxin. We have Clostridium. We also have perfringens. Okay, perfringens. Now, so we have another C, which is Corynebacterium diphtheria, which releases a diphtherium toxin. But this is for another lecture. Diphtheria. All right. Clostridium, listeria, bacillus thuringiensis. This would be for next time. So now we also have branching filamentous rods. So these kind of bacteria, they form branches. So this is branching filamentous rods. Under the branching filamentous growths, we only have two. We have the actinomyces, and then we have the nocardia. Okay. Then our main topic for today, which is the cocci. So for the cocci, we have we can divide this into the catalase positive and the catalase negative. Okay. So catalase positive, and then we have catalase negative. So under catalase positive, you see, it's better to group this because when you group it, it can help you to answer so many exam questions. So when it comes to catalase positive, we can also subdivide this into coagulase positive, and coagulase negative. Okay, so under coagulase positive, we have staph aureus or staphylococcus. So the S is staphylococcus, staph aureus. And then under coagulase negative, we can also subdivide this, right? So for this, under this, we can subdivide this into novobiocin. So we have novobiocin sensitive. I'm just gonna put in a negative for novobiocin negative. So for novobiocin positive, we have S epidermis. And for novobiocin negative, we have S saprophyticus. So on the exam, there is this clinical veneer, there is a question stem, right? The question would be like, a patient presents into the clinic room. The patient has osteomyelitis. The patient presents in the clinic room with osteomyelitis, and the patient also has some form of arthritis. All right, and then they tell you that it is a gram positive. The patient is infected by a gram positive, catalase positive, coagulase positive organism. Which organism are we talking about? It's Staphylococcus aureus. So S aureus is a gram positive, catalase positive, coagulase positive cocci. And then S epidermis is novobiocin, coagulase negative, catalase positive. Okay, just drawing this table or this diagram should allow you to answer certain questions on the exam. All right, now when it comes to the catalase negative, we can divide this into different parts, 3 categories. So so the first one is in terms of hemolysis. All right, so we have alpha hemolysis, beta hemolysis, gamma hemolysis. So that is how we're gonna do this. So so the first one is alpha. Let me put this here. So this will be alpha hemolysis, this will be beta hemolysis, and this will be gamma hemolysis. So for the alpha hemolysis, it's partial, so it usually turns green. Alpha hemolysis is partial and the color is green. Beta hemolysis is complete hemolysis, so it's gonna beta hemolysis is complete. And then gamma hemolysis, there is no hemolysis. All right, so let's move on. So when we look at the alpha, we can subdivide the alpha hemolysis, beta hemolysis, and gamma hemolysis. We can subdivide them into different bacteria. So the first one will be aptogen. So there is something called the aptogen, right? So we can have the aptogen positive, aptogen positive, and then we have aptogen negative. So the organism which falls under aptogen positive is S. pneumonia, which is Staphylococcus pneumoniae. And then we have S. viridans or viridan group. Forgive me if the spelling is if my writing is very small. So we have the S. pneumonia, and then we have the S or viridan group. So for the beta hemolysis, we have group A, strep, and group B. Oh, mind you, so the S here is for Staphylococcus epidermis, Staphylococcus saprophyticus, but the S here is Streptococcus. All right. So this is Streptococcus pneumoniae, Streptococcus viridan. And for the beta, we can think about group A and group B. So we have group A, strep, and we have group B, strep. All right, group B, strep. For group A, strep, it is called S. So group A is S. pyogenes. And the group B is S. agalactia. All right, so group A strep. And another way we can also separate this is bacitracin, because this is bacitracin positive, this is bacitracin negative. And for the gamma, we have the enterococcus. Like all the enterococcus groups, they fall under the gamma hemolysis, which is there will be no hemolysis, so it's gonna be so the next thing we're gonna talk about is we're gonna go into detail and kind of like go into each bacteria and talk about how they all look, some of the features, their mode of transmission and the features and stuff like that. Okay, so the first one we're gonna start with is the Staphylococcus aureus. All right, so the S. aureus, which is very, very common. It's one of the most commonly tested bacteria on the exams, the S. aureus. So the first thing is, where does it colonize? So they colonize the nerves. So the nose, the axilla, and the groin. All right, so they will colonize the nerves or the nose, the groin, and the axilla. All right. Remember, these are high yield points. You just want to focus on the high yield points. So they have, so every bacteria, they have like you know how we have resistance. Bacteria, they always develop some form of resistance against drugs and stuff like that, right? So most of the time when we use antibiotics on these bacteria, they also develop some form of resistance against the antibiotics. So next time we're gonna be talking about antibiotics. So there is like, we have antibiotics for most of the different bacterial infections that we have, but for this for the benefit of this lecture, let's just focus on like their mode of transmission, where they affect in the body, and some of the symptoms that we see when we are affected by these bacteria, so that we can focus on antibiotics on an entirely different lecture. Okay, so when someone comes in with Staphylococcus aureus, the first thing you're gonna see is that they will be they attack the nose, the groin, or the axilla. So for some of the symptoms, all right, They are the most common cause of osteomyelitis. So most common cause of osteomyelitis, and septic arthritis in adults. Septic arthritis. They can also cause scalded skin syndrome. Scalded skin. You need to like memorize these diseases. Like every time you hear scalded skin, always think about Staph aureus. Okay, scalded skin. They also cause toxic shock syndrome. This usually occurs in women who use tampon. Okay, so it's usually associated with tampon use. You end up with toxic shock syndrome. So their virulence factor is protein A. Okay, so their virulence factor is protein A, and that is one thing that you see among all the different bacteria. They all have virulence factor, which is something that they also use as a means of protection. Okay, way that they can also conquer the body. So they have protein A. What does protein A do to the body? So protein A usually binds to it binds FC portion. Let me just write FC. So FC, if when we talk about, when we start talking about immunology, we're gonna talk about the antibody. Okay, so when we look at the antibody, we have the FC, which is the constant region of the antibody, and that is where the Staph aureus, that is where they attach to, and then used as a means of their virulence factor. So yeah, so protein A goes to FC portion of the immunoglobulin. That's one thing you want to think about. So based on what we've done so far, you have an exam question, right? And then a patient comes into the hospital. The first thing the patient comes in, the patient presents with scalded skin syndrome. There is some form of osteomyelitis. There is septic arthritis and stuff like that. Okay, you think about Staph aureus. So high yield points, they attack the nerves, groin, axilla, osteomyelitis. The most common cause of osteomyelitis and septic arthritis in adults. They cause scalded skin syndrome, toxic skin syndrome, toxic shock syndrome, which is usually due to tampon use. And then we have the protein A, which is their virulence factor. How can we treat this? We can, they also cause, they are usually found in people who use drugs. You know, those people who use drugs intravenously. So when you inject drugs into your system and stuff like that, you are more prone to getting infected with Staph aureus. So we can also add this here. So IV drug users or intravenous drug users. They can also cause endocarditis. How do we treat this? We can treat this with vancomycin, which is one of the antibiotics we're going to be talking about. So we can treat with vancomycin. So this is all about Staph aureus. These are like the higher points. Trust me, if you know this, you're gonna get every Staph aureus question right. Why? Because besides knowing this, you also know it's coagulase positive, you know it's catalase positive, you know it's gram-positive cocci, you know it's blue or purple in color, you know it's made up of teichoic acid, you know it's made up of thick peptidoglycan layer. Bro, like, you know all this, you're gonna get every Staph aureus question right. Okay, now let's move on to the next bacteria, which is Staph epidermis. This is actually novobiusin positive. It is part of the normal skin flora. Okay, so this is a normal skin flora. All right. What's another interesting thing? We see this in infected. It affects orthopedic joints. Okay, Yeah, so it usually affects joints, orthopedic joints, ortho joints. This can also cause endocarditis. Endocarditis. It can also cause endocarditis. It's associated with the use of biofilm. You see how, like, for Staph aureus, it's usually common in people who are IV drug users. This is also commonly found in people who use biofilm. So, biofilm use. All right, so how can we treat this? We can also treat this with vancomycin as well. We can treat this with vancomycin. Then we have S. saprophyticus. To be honest, this is why here. Based on all the practice questions I've done and all the exam questions I've done, most of every question I get on this is like the fact that it is the second most common cause of UTI. Okay, it's like it causes UTI in sexually active females. So when a female presents into the hospital and the female is infected with UTI, the first you want to think about is E. coli. E. coli is like the #1 cause of UTI in sexually active females. The #2 cause, you think about saprophyticus. So on the exam, like every saprophyticus question is going to be about UTI. It's commonly found in sexually females. So let's put it here. So sexually active females. So it causes UTI in sexually active females. All right. Let's put this here that this is thick peptidoglycan. And then we said it's also made up of the teichoic acid. So on the exam, patient presents into the hospital, patient has UTI, and they did the lab work and they realized it's a gram-positive bacterial infection. Don't think about E. coli because E. coli is gram-negative. If you, when it's gram-positive, you think of Staph saprophyticus. A patient presents into the hospital. This patient is involved with using catheter tube or biofilm. The person presents with some form of endocarditis. You think about Staph epidermis. Okay, the person comes in with scalded skin syndrome, toxic shock syndrome with cause of tampon use, and stuff like that, osteomyelitis, septic arthritis. The person is an adult. You want to think about Staph aureus. Okay, so these are the gram-positive, catalase-positive organisms. Now let's move on to the catalase negative. So we actually gonna the first part is gonna cover this whole part from the gram-positive cocci to, and then we will see if we can touch on actinomyces and nocardia if we have time. But I'm just trying to make sure that the video is not too long, because there are some people when they see a video and it's like an hour long, it's kind of like discourages them and stuff like that. So when we break it into 2 parts, then we cover 1 part and then another part in the next video. So the first thing we want to talk about is pneumonia. Pneumonia. So streptococcus pneumonia or pneumonia. The first thing we wanna think about is the main virulence factor. Okay. So the virulence factor will be polysaccharide capsule. Polysaccharide capsule. So what did I say? For Staph pneumonia, the virulence factor is polysaccharide capsule. Okay. We know it's a partial alpha hemolysis is aptogen positive. So let's see. So now let's talk about the diseases that they cause. There is a very interesting mnemonic you can put there is MOPS. MOPS, MOPS. So the M is for meningitis. M is meningitis. Okay, the M is meningitis. The O is otitis media. Otitis media, which is a form of ear infection. Otitis media. The P is for pneumonia. I mean, from the name, you know that it's definitely gonna cause pneumonia. And the S, the last one, the S is it causes. Let's see. Why did I forget to? Okay, so inflammation of the sinus. So sinusitis is for the S. Next time I'm gonna blend in the colors instead of like using black for everything. I'm gonna try to put red and blue and stuff like that, so we can kind of like be able to distinguish like the bacteria and the symptoms and stuff that they are causing. Okay. So yeah. So S pneumonia, like the only thing you wanna think about, you know, the high yield points are: one is aptogen positive, is alpha hemolytic, catalyst negative, gram positive. You think about the virulence factor, which is polysaccharide capsule, all right? And then the symptoms or diseases that they cause, you think about MOPS, like mopping the floor, MOPS, which is meningitis, otitis media, pneumonia, and sinusitis, which is inflammation of the sinus. Then we have the viridans group. So the first thing you want to see is it's most likely the opposite. You see how this is aptogen negative, positive. S viridan is aptogen negative. Okay, this one is capsulated. As pneumonia is capsulated, they have the polysaccharide capsule. This one, the viridan group, they are not capsulated. So not capsulated or not encapsulated. They are not encapsulated. The most common high yield you need to know about the viridan group is dental caries. You know, when you see a question and then there is a bacterial infection and the person ends up with dental caries, think about streptococcus viridans. Trust me. Dental caries. If there is anything you want to know about the viridan group, aptogen negative viridan group, dental caries. Just like the same way you associate saprophyticus with UTI in sexually active females. Yeah, it's the same thing. And then we have the group A strep, and then we have the group B strep. Okay, now let's start with the group A strep. The group A strep is the Streptococcus pyogenes. The Streptococcus pyogenes. Let's see. 4. So they have a capsule. So this one is capsulated. So they have capsule. All right, that's the first thing you want to know. And their virulence factors. We have the M protein. There is streptokinase. There is streptolysin O. If you can't see it, when I say it, you can write it. Okay, so the virulence factors for group A strep, the first one is M protein, second one is streptokinase, the third one is streptokinase, we have streptolysin O, and then we have DNases, which breaks down DNA, so DNases. All right. How do they cause? We call them the Jones syndrome. The Jones syndrome. So you think about group A strep, think about Jones. I don't know if you know any Jones. Think about any Jones you know. If there is any movie, Indiana Jones, when you think about Indiana Jones, think about the fact that he has a group A strep, or he's suffering from streptococcus pyogenes, the Jones syndrome. So the Jones syndrome, the first one, the J is for joints. All right. So when a person comes to the hospital and the person is infected with streptococcus pyogenes or group A strep, they're gonna be experiencing joint pains, so it's gonna attack the joints. And the O here is for the heart. All right, it's for the heart. So it causes heart problems. Okay. The N for the Jones is nodules, which are found on the extensor surfaces. And then we have E, which is erythema. Reddish discoloration of it's more like ecchymosis and stuff like that. So erythema, and then the S is Sydenham's chorea. Let me write this. Sydenham's chorea. All right, so let's quickly go over this. Staphylococcus pyogenes, they are encapsulated. They are virulence factors: M protein, streptokinase, streptolysin O, and they have DNases, which also helps to break down DNA. And then the symptoms, or their clinical symptoms, we have the Jones. The J is for joints. The O is for heart problems. The N is nodules, which are found on the extensor surfaces. We have the erythema, and then we have Sydenham's chorea. These are all the key points, like the higher points you need to know about group A strep. Now let's talk about group B strep. Oh, and remember, group A strep is bacitracin sensitive. Group B strep is bacitracin resistant. Okay, that's another key point you want to add to your notes. All right, so group B. What helps me with group B, which is agalexia, what helps me is the fact that it's group B. So when I think about group B, I think of babies. So it's the major infection in newborns. So this is the major infection in newborn babies. Trust me, when you have an exam question, and there is a newborn baby which has been affected with any form of bacterial infection, the first thing you should be looking for is group B strep. Okay, and they are transmitted through the vaginal canal. Okay, so their mode of transmission through the vaginal canal. All right, so what are some of the things? So they cause sepsis in newborns. So sepsis. So they cause sepsis, they cause pneumonia in newborns. Pneumonia. All right. Virulence factor, they are made up of capsule. So they are made up of polysaccharide capsule. Okay. All right, so these are the higher points you want to know about the group B strep. Babies, group B, babies, transmission, vaginal canal, babies, newborns, group B, babies. And then they're gonna end up with sepsis in the newborn baby. They're gonna end up with pneumonia. You can treat a person with penicillin. You can, treat with penicillin. But like I said, I don't want to go into crazy detail. I don't want to talk about antibiotics here, because we're gonna have separate video for antibiotics, and for every antibiotic we're gonna be talking about vancomycin, ceftriaxone, and all that. We're gonna be cephalosporins, and all that, penicillin. We're gonna be talking about that on a separate video. But for now, let's just focus on the virulence factors and the symptoms of these bacterial infections. Now, the last one we wanna talk about is enterococcus. Enterococcus. For enterococcus, let's see. We can also divide this into faecium and fecalis. All right, fecalis, and then enterococcus, faecium. All right, so we have fecalis and faecium. Fecalis is more common compared to the faecium, but they usually end up causing something like similar diseases and stuff like that. When someone presents with enterococcus, chances are the person is gonna present with some form of endocarditis or UTI. Okay, so some of the clinical features we have endocarditis. We can end up with UTI. Endocarditis, UTI. Another high yield point. All right, they grow in a medium of sodium chloride. So when you put them in a medium containing sodium chloride, they grow up to 6.5% sodium chloride. All right, that's another high yield point. You know, so they grow in a medium. Up to 6.5% sodium chloride. That's it. Now let's quickly go over this. So the first thing we spoke about gram-positive. We said gram-positive bacterial. The first thing you want to talk about is their thick and peptidoglycan layer. The next thing you want to distinguish them from the gram-negatives is the fact that they have teichoic acid. Okay. We can also think about the fact that they stain blue or purple. Okay. Now we divided the gram-positive. We divided them into different groups: rod, branching filamentous rods, and then we have the cocci. For the cocci, catalase positive, catalase negative, catalase positive, coagulase positive, coagulase negative, coagulase negative, novobiosine positive, novobiosine negative. Just go over the video. If I'm missing things, this can help you to create this blueprint or template. You can also use this as a template to fill in all the things that you deem relevant as a high yield and put it in yourself. Okay, but pretty much if you know these features of the gram-positive like we've listed here, it should help you to eliminate all the wrong answers. Because right now we've seen UTI twice, right? We've seen UTI in enterococcus, we've seen UTI in staphylococcus saprophyticus. So the person comes with UTI, you can't just say E. coli, saprophyticus, enterococcus. You also need so a person comes into the hospital, the person presents with a UTI, right? And then they do lab culture, and then they realize that the bacteria grows up to 6.5% in sodium chloride. You're not gonna think about E. coli or saprophyticus. You're gonna be thinking about enterococcus. The person presents into the hospital, the person is novobiosine. They did lab work and they realized the bacteria is novobiosine sensitive or resistant, novobiosine resistant, and the patient is a sexually active female. What do you think about saprophyticus? So this will help you and lay out the blueprint for you to eliminate most wrong answers and choose the right answer. All right, so this is the first part. The second part, or the next video, we're gonna be talking about the branching filamentous rods, which is actinomyces nocardia. We're gonna also talk about the bacillus groups, Anthracis cereus, Listeria. We're gonna be talking about Clostridium species, the Tetanus botulinum perfringens. I think there is one more, and then we have the C. diphtheria. We're gonna talk about all that in the next video. Thank you for watching this video, and if you have any question or any comment, if I made a mistake or anything, please make sure you put it under the comment section so I can see it and then make any corrections where I deem it relevant. Thank you, and see you in the next video. Take care. Bye bye.